All right, these are the notes for the viral structure and reproduction section. Within this section, we will be looking at 3 different types of viruses: enveloped virus, the helical virus, and the polyhedral virus. We'll look at the enveloped virus first. An example of an enveloped virus is influenza. You can see that the enveloped virus has a capsid that is enveloped by surface proteins. Inside of it, we see the nucleic acid along with the lipid envelope on the very outside. If we move on to the next type of virus, this is the helical virus. An example of that one is rabies. The structure of this one does not resemble that of the enveloped virus. A helical virus is going to have its nucleic acids along with its capsids in a helical form inside of a surface proteins and lipid envelope. Lastly, we will be looking at the polyhedral virus. An example for the polyhedral virus is that of foot and mouth disease. It gets its name from the shape on the outer surface proteins, but we can see the capsid and the nucleic acids inside of it. All right, now that we've seen 3 different types of viruses, we can look at this general virus just so that we can notice different structures that make up the virus. So what you see here is a bacteriophage. It infects bacteria, right? And this bacteriophage, much like other viruses, is made up of 4 different components. We have the DNA inside. That was referred to above as the nucleic acids. Remember, nucleic acids are the monomers of DNA. The DNA is protected by a capsid. Much like a head, it protects the DNA. Below the capsid is the tail sheath. And below the tail sheath are the tail fibers. All right, we are going to be transitioning into the reproduction of a virus, but before we can go over that, we have to discuss how a virus enters its host cell. So a virus enters a cell in different ways. If we look at bacteriophages, remember bacteriophages infect bacteria. These pierce the host cell, as you can see in this image, where the viruses are painted in a neon green and the bacteria is almost a rusty orange color. Here's another image. You can see a more defined look of the tail sheath and the tail fibers, as well as the capsid, and even the DNA of each of the viruses on this bacteria. All right, viruses that attack eukaryotes, not prokaryotes. Remember, a bacteria is going to be an example of a prokaryote. Viruses enter eukaryotes via endocytosis, which is an invagination or a way in which the cell devours things from its outside environment. Viruses that attack eukaryotes must fuse with the membrane. Here we see a eukaryote. The first thing you should notice is that within the cell, within the eukaryotic cell, there's much more organization. On the bottom here, we can see the nucleus with some nuclear pores. Now, RNA is leaving the nuclear membrane, and you can see that there's some communication between these viruses on the surface of the eukaryotes and the nucleus, and we'll get to that shortly. All right, so this is the last portion of your notes. We are going to go over two different types of reproductions, or two types of infections. The first one we'll go over is Oolitic Infection. This is going to lead to the host cell death. It starts where the bacteriophage attaches and injects its DNA into the host bacterium. Then the viral DNA forms a circle. You can see the circle in blue. After this, the viral DNA is going to direct the host cell to produce new viral parts. The parts assemble into new bacteriophages, as you can now see inside of the cell. And finally, the host bacterium is going to break apart. You can see the cell membrane is going through lysis. Bacteriophages are able to infect new host cells. All right, the other form of infection starts the same. The bacteriophage attaches and injects its DNA into the host bacterium. The viral DNA forms a circle, at which point we go down here All right, at which point we go down here. So the viral DNA is called a prophage when it combines with the host cell's DNA. So in this case, the circle, which is still in blue, is the virus's DNA, the viral DNA. This host cell's DNA is in red or pink, and it has combined with the virus's DNA. After this initial point, the prophage is not active, and it continues to replicate, passing on both the host cell's DNA and the viral DNA into its offspring. Many cell divisions occur, and they produce a colony of bacteria that have been infected, but it does not lead to the host cell's death.